All right, let me go ahead and get started. Mark, I apologize. This probably won't be the best introduction because I wasn't prepared to be the one hosting this, but everyone within the ELK community who's here today, glad you could join us for this presentation from Dr. Mark Musin. I think probably most of you know Mark, especially if you have been anywhere in the ontology or standard space. But Mark is at Stanford. He's a professor of biomedical informatics research, medicine and biomedical data science. He is director of the Stanford Center for Biomedical Informatics Research, and he conducts research related to open science, data stewardship, intelligent systems, and biomedical decision support. He has been elected to the American College of Medical Informatics, the Association of American Physicians, the International Academy of Health Sciences and Informatics, and the National Academy of Medicine. So that's where I'm going to end talking, reading off his page. More importantly, at least I know Mark because we use many of his products and tools that he's developed over the years. In particular, he's PI of the National Center for Biomedical Ontologies, and if you are interested in finding ontologies, this is the place to go. And he has been instrumental in leading biomedical work in the biomedical ontology space forward. He is principal investigator on the Center for Expanded Data Annotations and Retrieval, known as CEDAR, which produces the CEDAR metadata toolkit, which we actually my group is using, has just recently moved over to adopting, and we're now using it quite extensively for our work in the climate change area. And his group developed Protege, which is really the tool if you're in the space of managing terminologies and ontologies. So Mark has done quite a bit of impressive stuff in the space that ELK cares about. I just see, Maria, you just got on. I gave a brief intro to Mark. Do you want to give any other comments?
I will just remind people of house rules, and I hope my internet does not drop again. Okay, share.
All right. You're going to do house rules?
Yes.
Okay.
Hope that's it. Yes, I'm still on. Sorry, that was very too exciting. I suppose you already reminded people about the mission of Environmental Health Language Collaborative, which is to advance integrative environmental health research by promoting access, use, and harmonization of data through interoperable terminologies and best practices. House rules are very simple. Please update your Zoom name to your first and last name and affiliation. If you have any questions for the presenters, please use the Zoom Q&A function or raise your virtual hand to speak. Please remain muted unless you're speaking, and this webinar will be recorded and posted to the ELK website. A link to the recording will be sent via the ELK listserv. If you want to learn more about ELK or become involved, you can visit our website at nhs.nih.gov.research.program/ELK. But it's simply just to Google ELK and NIHS. You will get to this page very fast. You will find our ELK resource catalog. You can also join the ELK email network by clicking this link on the web page. If you have any ideas about new use cases or semantic needs, ELK activities or training, new ELK activities or training, if you want to add some useful resources to our portal, please don't hesitate to contact us directly by emailing either elk@icf.com or myself directly or Charles. With that, we can move to our presenter today, which you already heard his very impressive bio, I suppose, even abbreviated. And the topic of the talk for today is Center for Expanded Data Annotation and Retrieval, CEDAR, Enhancing Open Science Through Standard Metadata. And with that, I will stop sharing my screen And Mark, please take it away.
Thank you, Maria, and thank you, Charles, for those kind words. I'm really honored to be invited to speak with everybody today. I'm going to give you an overview of the work that we're doing at Stanford in the space that's important to this community in terms of being able to declare standards for data, to share data, and basically the kinds of technologies that I think are going to be needed down the road in order for open science really to take root. We know that about 10 years ago, the world kind of suddenly took notice when a group of investigators got together and published the FAIR guiding principles for scientific data sharing. And this paper had an enormous impact. Everybody wanted his or her data to be more fair than the next person's. And the problem is most people actually don't really look carefully at what the guiding principles are and what we need to have fair data in the first place. When you look at the principles, they're kind of overwhelming because there's just so many of them. But I'll make your life simple. When you look carefully, almost all of them are about metadata. And actually, when you look even closer, you see that they talk about the need for rich metadata, for richly described metadata with a plurality of accurate and relevant attributes, metadata that meet domain relevant community standards. These are the kinds of things that we need for our data sets to be fair. And I'll tell you, unfortunately, most of the metadata in the world don't meet the FAIR principles in any way. Metadata in our public repositories actually are pretty terrible. Most investigators view their work as publishing papers. And the idea of leaving a legacy of data for someone else to analyze is just not part of the culture. And although now sponsors and managers all require data sharing, there's a hesitancy to use grant funds or project funds to pay for it. And when you look at what's in repositories, what you see is that our data sets are hardly AI ready. I won't say this is a random metadata specimen from Biosample, one of the NCBI data repositories. But you can see that the kinds of descriptors on the left-hand side are pretty arbitrary. Things like cell type, I'm not sure what a cell type is, not what a cell subtype is. What it means for a cell type to be primary cell is not clear to me, and I don't think anybody would ever search for the term primary cell. And then you see really unbelievable stuff like ethnicity Asian, or worse, race, yellow race. Here's another data set where the isolate is missing apostrophe, age, missing apostrophe. These are not the kinds of things that anybody would ever search for. Here's just another kind of metadata, another sample where the sex is given as W, which I guess is woman, but I guess that's not what anybody would search for in looking for sex. And you see lots of duplicates, lots of real garbage, and then another example here. There's an attribute called hereditary way. I don't know what that means. I think it sounds to me like an address. Altitude. I never knew that a data set had an altitude. And start and ends that really don't make any sense. The problem is when you look at Biosample, it's really quite unbelievable. And Biosample is viewed as one of the best NCBI databases. 73% of the metadata values that are supposed to be Boolean are not actually true or false. They're values like non-smoker or former smoker. 26% of the values that are supposed to be integers don't parse as numbers. They parse as just absolutely bizarre strings. 68% of the metadata entries that are supposed to represent terms from ontologies are not terms from ontologies. Presume normal, wild type with an underscore. I mean, we're in a situation where if people want to search for metadata, if they want to be able to put data sets together, if they want to analyze them using AI technologies, we're in bad shape because the descriptors for our data sets do not lead to fair data, and our data really don't meet the requirements of data sharing and data reuse. What I want to do this afternoon is talk about what we need to make our data sets fair. And what we really need, in my view, is to have ontologies that provide the controlled terms that standardize the metadata that are used to describe data sets. We need reporting guidelines that provide a standardized structure for metadata components. We need technology to make it easy to author good metadata in the first place. And we need procedures that allow community-based standards to be encouraged, to be used, and we'll talk about that as well. So it's basically a function of ontologies, reporting guidelines, technology, and procedures. And I'll talk about these in turn. We'll start with ontologies, which I know ELK has obviously spent a lot of time worrying about. Go to a repository like the Gene Expression Omnibus, and Geo is a mess. If you want to know the age of a specimen, you see age and age with a capital A and age in all caps and pop age and age after birth, and just dozens of ways of expressing age. If you're an investigator and you want to search for a data set that deals with subjects of a certain age, you have to anticipate all the ways in which an investigator might have expressed the term age, and you have to anticipate all the possible misspellings, all the typos. It just becomes really hard. In fact, my colleagues who are really interested in being able to do data reuse have teams of graduate students who manually sort through data sets because search just doesn't work when you don't have standards that enforce agreed upon ways of expressing things. At Stanford, we spent a lot of time creating the BioPortal repository, which basically is a way of finding all of the publicly available ontologies in biomedicine. And when you go to BioPortal, you can see the kinds of ontologies and control terminologies that are available in biomedicine. You can drill down and you can see the metadata for ontologies, like in this case, the foundational model of anatomy. You can see who owns it, who maintains it, how many versions there are, how it gets used. And then you can actually browse through all the terms, get a sense for how the FMA describes anatomy and understand how it all fits together. And I think it's really important for us who care about data sharing and care about making data sets AI ready to have this understanding that ontologies are critical in describing the metadata that define our data sets. But it's more than just ontologies. And I think we forget that we need more than control terms. We need reporting guidelines. We need a standardized structure for making sure that our metadata mention all the things about the data set that are relevant and that are going to be expected by someone who wants to find a data set and use it for secondary analysis. The good news is that we're doing a lot of this in biomedicine already. 25 years ago, the microarray community recognized that it really needed to have standardized metadata so that people would understand when one did a microarray experiment, what was the substrate, what kind of chip was used, what were the experimental conditions. And the result, as you probably know, is Miami, the minimal information about a microarray experiment. And Miami initially was just a checklist of all the things that the metadata needed to contain. But we can think of Miami and all of its successors as ways of standardizing what's in a metadata description. And in biology, we have minimal information about T-cell assays, minimal information required in the annotation of biochemical models, MIATA, Miriam, Minimes, MISFISHI, and we have really hundreds of these standards. And I think this is exactly the kind of thing that we need more of, particularly in environmental medicine. So we need not only the ontologies, but we need to be able to say, in addition to the ontologies, we need to have a structure that tells us what are the descriptors that belong in the metadata, how can we define standardized ways of talking about data sets so that we can be sure that a metadata record includes all the things that need to be said about a data set and that we say it in a standardized way so that we can search for it. Whoop. So I said we need, in addition to ontologies and reporting guidelines, technology. And that's really what my group at Stanford has spent a lot of time working on. I'll tell you about two kinds of technologies we've created. One is the CEDAR Workbench, which is an editor that helps investigators to create standards compliant metadata in the first place. Basically, it's an editor that allows us to use ontologies and reporting guidelines stored in a library so that we can create standardized and formal descriptions of metadata. And also talk about something which is relatively new, so the metadata validator, something that we're using in the context of the Common Fund's HubMap project, which allows us to deal with metadata that are not authored using CEDAR, that are entered more ad hoc using spreadsheets, and at least validate those metadata to make sure they adhere to the appropriate metadata guidelines and ontologies. Let's start with the CEDAR Workbench. The CEDAR Workbench basically can be viewed as a method that includes technology for authoring metadata templates. So we assume that we're going to take a reporting guideline for metadata, and we're going to store that not as the kind of text that you would see in a checklist like Miami, but rather as something that's computational, something that a computer can parse and understand. And that means a template that's going to have information about the descriptors and the values of those descriptors for a metadata record. And the template basically then becomes the basis for what you see in the middle panel, which is basically a metadata acquisition system, which allows us to use the template, which describes the reporting guideline, which allows us to use ontologies stored in the bioportal repository that I mentioned, and to fill in the metadata form derived from that template in a way which allows us to actually have metadata that adhere to the standard. And we create a local cache where we have that standard available to us. But most important, we can use the metadata that we've created with the metadata editor as a basis for the metadata that are used in repositories like Geo or Import or a variety of other repositories that people use. So here's what CEDAR looks like. What you see is a library of templates where each template corresponds to a reporting guideline, where that reporting guideline is basically, as I said, the expected terms that are going to be used to describe a data set in that particular discipline or for use with a particular repository. And you can say, okay, I want to select a template from this library, and I might want to populate it. So I'm selecting the biosample template, which is a template that allows us to create metadata that are going to go into the NCBI's biosample repository. And what we see are a set of attributes. These are the standard attributes from the data dictionary that biosample uses, not unfortunately what most biosample metadata records actually use, but the data dictionary suggests that a biosample record, particularly one that deals with a human subject, has to have a sample name, an organism, the tissue, the sex, the isolate, the age of the subject, biomaterial provider, and so on. And we can fill these in. We can say the sample name is 56, the organism is Homo sapiens, for example, and the tissue is, in this case, selected from a drop-down list. Where does that drop-down list come from? It comes from an ontology or a controlled terminology that the template developer has specified. And in this case, that means the Uberon ontology, which is stored in Bioportal. And so the tissue could be blood or liver or bone marrow or whatever, and the user makes a selection. The important thing to stress is that we don't present to the user the hundreds of tissues that are actually in the Uberon ontology, but rather we see them sorted so that the most likely selections are going to be at the top. Because remember, we keep a local record of every previous metadata entry that's been entered through CEDAR. And we know when someone is filling in the biosample human template, that most of the time the tissue is going to be blood. And so that percolates to the top of the list in the drop-down menu. And the user then makes a selection. The thing to stress is that this form that you're seeing for entering metadata is not canned. Basically, is created on the fly by looking at the metadata template stored in the library. The template tells us what are the attributes. It tells us what are the ontologies to use. And so what you're seeing here is a web form that's generated automatically based on the information that's stored in the CEDAR library. And so this gives us a way of automatically generating a mechanism for entering metadata, but not just any metadata. Metadata for biosample human in this case that conforms with the data dictionary, that adheres to the standards, and avoids the problem of entering the kinds of junk that I showed you in all those other biosample metadata records a while back. What makes us very excited is that not only is CEDAR being used in a wide range of NIH-sponsored projects, but it's being adopted elsewhere. So for example, Dryad is one of the generalist data repositories in the NIH GRAY initiative. lots of people use Dryad, not just for biomedical data, for all kinds of scientific data. And Dryad has recently incorporated CEDAR so that people who use Dryad can have access to CEDAR templates and use those templates to create standardized metadata for whatever mechanisms, whatever disciplines are relevant for the data that's being entered. And so what makes it really nice for us is that people in a variety of specialties can use Dryad for storing their data, and they get CEDAR almost for free. And they have the mechanism for creating CEDAR compliant templates and therefore standardized metadata for the data that they put into the Dryad repository. The Open Science Framework, or OSF, is doing the same thing. So here's the template repository in OSF. These are, again, CEDAR templates. Users of OSF can select the template required for their particular area of study, and they have a web form embedded within the OSF resource that allows them to fill in the standardized values for the standardized fields, just as I showed you earlier. And that's really pretty exciting for us as well. And as well, OSF has created a mechanism in their app for accessing CEDAR. So this is actually for us the first time that our work has been actually made available through an app. And again, if you're using the OSF app on your iPad, you can get access to standardized descriptions of metadata and make sure that the data that you enter is compliant with the reporting guidelines that your particular discipline requires and the ontologies that are specified in that reporting guideline. So that really was the CEDAR workbench. And so basically the CEDAR workbench gives you the mechanism of having a library of standardized reporting guidelines and ontologies, which can be used by investigators to ensure that the metadata that they use to describe their data sets adhere to the appropriate standards, something which is really hard to do without having this kind of technology. What I want to talk about next is a different technology system, the CEDAR Metadata Validator, which basically allows us to validate spreadsheet-based metadata. We are very active in HubMap, the Human Biomolecular Atlas program, which is, as I said, a common fund activity. The whole goal of HubMap is to come up with an atlas that will describe the biomarkers that allow you to understand what characterizes basically every kind of cell type in the human body. And we were very excited when HubMap asked us to work with them. HubMap really has religion about standardizing metadata and using standardized ontologies. And we've worked with HubMap to create about 36 templates for descriptors, in this case, reporting guidelines, for each of the kinds of assays that are being used in HubMap. And what we have is the ability for CEDAR to provide the mechanism for filling in the blanks through a web form, just as I showed you earlier, for each one of these types of assays, so that, again, the investigators at HubMap can ensure that their metadata are compliant with the standards that HubMap has created. That's really wonderful for us, and we're really excited about that. And we were really thrilled at the possibility that we'd be using CEDAR and HubMap. But we learned that we weren't going to be successful. HubMap investigators just did not want to adopt a new technology. They were not interested in filling out web forms. They love spreadsheets. And the way that they've become used to creating metadata is by dealing with the fact that they have multi-value metadata because they're dealing with complex assays of multiple specimens at once. They want to put their assay metadata in spreadsheets. They don't want to deal with anything else. And so we had to figure out what to do with spreadsheets. So the problem, of course, is that spreadsheets don't easily enforce standards. They're prone to errors. They often have missing fields, formatting problems. Validation features in Excel are very limited. And the whole metadata ingestion process for HubMap was in a situation where it needed to be able to rectify errors in the spreadsheets, but didn't have an easy way to do that. Well, our solution was basically to take the idea of CEDAR templates and to use CEDAR templates as the basis for facilitating a validator for the kinds of data sets that were being entered into the HubMap program. So basically, we could take a template out of CEDAR's library and use that to inform a system that would look at the spreadsheet and determine whether the metadata entered into the spreadsheet actually were adherent to the guidelines that HubMap wanted to enforce, looking both at the reporting guidelines and the ontologies. So here's what a spreadsheet would look like. As you can see, it's kind of difficult to just inspect the spreadsheet and make sure that everything adheres to standards. But what we do is we ask the investigator to take the spreadsheet and just drag it into a box in this spreadsheet validator system and push a button. And what we will do is automatically use the CEDAR template that corresponds to that spreadsheet and offer the user a summary and say, look, there are some errors. We know that actually 13 out of 20 entries here are okay, but there are obviously a lot of errors that are not okay. And we can say, look, some of those errors are missing values. So there are places where you had to enter a value for the preparation condition or the storage condition or the index number or the sample ID. Those are missing values. Those have to be added. There are values where the value types are incorrect, where you presumably needed to use a term from a predefined value set or an ontology, and you didn't do that. And so this kind of feedback can be very valuable. But we do more than just provide the feedback. We actually have a mechanism where we can use the CEDAR template and say, look, we'll do our best to try to fix the spreadsheet on our own. We can try to identify where there are values that are not standard terms, where you have entries that are supposed to be numbers that are not numbers, where you have values that are not strings. And basically, we can automatically correct to the extent possible the errors. And the result is that we have the ability to take the spreadsheet and convert it to a form which adheres to standard much better than we can with just dealing with spreadsheets on their own. So basically, we have two kinds of technologies. We have the CEDAR Workbench, which is an editor where we can generate a web form from a template and use that web form to enter for an investigator to enter standards compliant metadata. And we have the metadata validator for investigators who don't want to use the web forms, who insist on using spreadsheets, but we can validate those spreadsheets again using the same kinds of CEDAR templates. Now, that's all good, but it's important to sort of take a step back and realize that the template is more than just something computational in a computer system. That template is a knowledge base. It basically serves as a way of encoding for the computer and for the scientific community what that scientific community's metadata preferences are. So it captures, if you will, in a reusable, standardized, computational form, what are the preferences of a scientific community regarding reporting guidelines for its metadata? What are the ontologies that it wants to use in those reporting guidelines? And people can use the template to review, enhance, and build on those preferences. And of course, machines that use things like the CEDAR Workbench and the CEDAR Metadata Validator can use those templates as well. So we have basically a knowledge base that is useful both to people and machines for encoding the preferences of a scientific community regarding how it wants to ensure that metadata adhere to appropriate standards. The question becomes, where do those templates come from in the first place? And basically, you have to build them. Over in the Netherlands, the Go Fair Foundation has been creating what they call Metadata for Machines workshops. And this is Go Fair's logo for these M for M or Metadata for Machines workshops. I take no credit for this logo. I don't know. I'm not even sure I know what it means. But basically, the Go Fair organization has really taken the lead in working with various scientific communities to identify what are the ontologies and value sets they need for their metadata, and what are the reporting guidelines that they need for their metadata to be complete. And basically what they do is they create workshops where they bring together scientists, put them in a room for a few days, and they basically tease out, if there are no existing reporting guidelines for their area of work, what do investigators expect in the metadata for data sets in their area of work? How can they standardize what are the fields of those reporting guidelines, so that we are sure that just as in Miami, we're saying what we need to say about the data set so people understand what needs to be incorporated so that ultimately we can search for data, we can integrate data, we can do AI. And basically, the result of one of these metadata for machines workshops is a new CEDAR metadata template that has the reporting guideline and the corresponding ontologies in a machine understandable form that, as I said, basically is a knowledge base that captures the community's preferences for these metadata standards. Now, what I think was also interesting is that there's a very close relationship between the GoPair organization and the Netherlands Organization for Health Research and Development, which in Dutch turns out to be something called ZonMV, which is basically the Dutch NIH. What ZonMV has done is that they have basically hosted a large number of metadata for machines workshops to develop metadata templates and control terminologies and value sets for a lot of the work that they're doing in the area of COVID. So they recognized early on that COVID as a public health emergency would require a lot of data sharing, couldn't tolerate annotation of data sets in a way which was confusing, and basically said, okay, they want to have CEDAR metadata templates for all of the studies that they're funding related to COVID. And what was really interesting about ZonMV is they said, okay, we're going to make it a requirement of our RFAs that basically the funding is predicated on a promise that investigators not only will obviously carry out the investigations described in their grant applications, but they will store their metadata in a public repository and that they will annotate their data using metadata templates that come out of these M4M workshops that were being created by the GoPair Foundation. And they're now looking at a number of other areas, particularly antimicrobial resistance and some other areas of science where they think it's really important for metadata to be standardized. And they're doing something which I think no other funding organization is doing that I'm aware of, and that is they're making it a requirement of funding that investigators actually adhere to metadata standards in addition to performing their work. So again, we can think of creating a metadata template through one of these M4M workshops as a situation where we have template modelers, people who really understand how to create a good reporting guideline, working with subject matter experts who know what are the expectations of people in a particular community of practice, creating metadata templates. And it's really been exciting to see the work in the Netherlands, as well as the work that I've seen not only in a number of NIH-funded activities, but also, as I said, in OSF and in Dryad, where the idea of being able to create metadata that adhere to standards really has taken root. So basically, we can look at CEDAR metadata templates and recognize that they're communicating metadata standards in a reusable fashion, that they're capturing the knowledge of a professional community in a machine-readable form, but also in a people-readable form. And basically, they can encode knowledge of what are the preferred standards that a community wants to use. And they help communities codify their preferences for metadata in a public, inspectable, editable place. And they ideally will be able to plug and play with a variety of software systems. And I've told you about the CEDAR Workbench, and I've told you about the spreadsheet validator. But I would imagine we could anticipate all kinds of technologies that would take advantage of CEDAR templates and work with them to ensure not only metadata compliance, but help one build new guidelines on the basis of previous guidelines, help identify where new ontologies need to be created, and so on. And basically, I think this kind of approach can help a particular community and also people outside that community to reuse knowledge basically over and over again. So I think we can think of a CEDAR template as a cartridge, if you will, a cartridge that can plug and play with a variety of applications that are needed to manage data in a variety of ways, and where the cartridges basically communicate preferences regarding community-based metadata standards. And I think we should be able to view what we're doing in CEDAR not just as a particular software system, but as an approach to standardizing metadata that I think has great generalizability, not only in biomedicine, but basically throughout science. So basically, making metadata fair requires good metadata. As I said, all the FAIR guiding principles deal with metadata, and they deal with metadata standards that are created by communities within scientific organizations. So really, what we need is to have more of this. We have a lot of emphasis right now on communities building ontologies. We don't have a lot of emphasis on communities building reporting guidelines for different kinds of experiments. And basically what we need is community endorsed metadata standards for the kinds of experiments that are done in science. We need technology like CEDAR that helps us create standards adherent metadata in the first place, and the metadata validator that helps us to improve metadata when investigators don't want to use a tool like CEDAR. And most important, we need to change the culture. We really need a concerted effort on the part of funders, publishers, professional societies, investigators, really to stimulate the creation of new standards to advance science. I'm really excited by what people have done in CEDAR, but most areas of biomedicine and most areas of science don't have standards at all. And there really needs to be a concerted effort on the part of all of us to work together to create the kinds of standards that we need so that we can create metadata that are searchable and findable and allow our data sets to be fair. And so really data sets are not going to be fair until funding agencies are able not only to create requirements for standardized data and standardized metadata, but they have to really enforce those requirements, which is, I appreciate, going to be difficult. Publishers are going to have to demand that metadata adhere to standards, and investigators are going to have to feel peer pressure if they don't create data sets that adhere to standardized metadata. Academic institutions are going to have to deem data sharing and the creation of fair data as an essential component of scholarship. I mean, just as we now expect publication to be a component of our evaluation in the academy, we surely are going to have to expect data sharing in a meaningful way to be a part of that promotion process as well. And really, we need mechanisms that are going to encourage professional societies to take the lead in developing community-based standards for their constituencies. Right now, professional societies in general do not feel that pressure. And it's okay for us to say that data sharing is a goal, but it's not going to be a goal until there really are stakes in the game. And so in the meantime, basically, it's just important for us to understand that we have semantic technology like CEDAR that can help us to make data fair, that can enable third parties to find and access other people's data. And basically, if we take advantage of the encoding of standards in tools like CEDAR, we do have the opportunity for data reuse. We do have the opportunity for making data fair. And I think that's really, for me, one of the exciting ways in which science is going to advance over the next decades. Let me stop there. Let's open the floor to discussion.
So there's a few questions in the Q&A. Can you see it, or would you rather me read it out loud?
I see the chat, and here's the Q&A.
Yeah.
Okay. With the validator, is it possible to define inter-element rules to handle more complex validation rules? Right now, no, but that's something that we would love to do. And so I'll say, as the usual grant recipient, with funding, anything is possible. But right now, our validation really is based on a review of each element in the guideline one by one. But I agree with you. What we really would love to do is look at more complex relationships, because that's where things definitely get interesting. How is the ontology information represented in the CEDAR system to enable, how is the ontology information represented in the CEDAR system to enable machine actionability and integration with other ontology-based systems? Well, let me answer that by saying BioPortal currently has virtually all the publicly available ontologies in biomedicine. And so we know that if we're taking ontologies out of BioPortal, we'll basically get the ontologies that we need for the kind of work that we do. We know that there are lots of things that are not in ontologies that need to be represented in value sets. And what we do in our work is we create new value sets or use existing value sets and put them into BioPortal so that we have a mechanism within CEDAR for accessing standardized lists of terms that are not part of standard ontologies. What we're really excited about is that BioPortal as an infrastructure has been taken up by a lot of other communities. So there are large groups that have used the foundational infrastructure in BioPortal and created ontology repositories in different areas of science. So for example, there's a group in France that has created an ontology repository for agronomy using basically using the BioPortal software. So you can go to agroportal.org and get agronomy ontologies. There's a group in Italy that has created something for ecology called EcoPortal. There's an Earth portal. There's a material science portal. And so what we're seeing are lots of people taking up what we've done in BioPortal and creating ontology repositories for other areas of science, which then could integrate with CEDAR. And so we're really excited about the possibility of using CEDAR not only for biomedicine, as we're currently doing, but through these other ontology repositories expanding its reach. And that's something that we realize hasn't happened yet, but it's something that we see down the line. Thanks for the compliment. Do you have an estimate of what proportion of US funded research is already captured in CEDAR? And I would say just a sliver. As I said, the problem is that most areas of science don't have fair metadata because they don't have metadata standards. And I think really what's necessary is not just a bunch of technology, but really to change the culture so that scientists, not only in biomedicine, but really in all areas of investigation, recognize that there's a need to be able to describe data sets in a standardized way, to create those metadata reporting guidelines, and to be able to use them in their work. I mean, I think biomedicine has had maybe a 30-year head start where people recognize that ontologies and reporting guidelines were important. But I don't think that's had the same kind of effect in other areas of investigation. And that's where we need a lot of work. And as I said, biomedicine certainly can claim a lot of victories, but we have vast areas of the work funded by NIH that really needs to pay attention to metadata, and where the metadata reporting guidelines and ontologies just don't exist yet, which I guess is why ELK is here.
Mark, I can't figure out how to ask the question, so I'm going to go live and ask it. I was thinking on that lines, you mentioned that different communities are starting to look at their own portals. Is there a way within CEDAR that we can mark or indicate certain templates are more appropriate for the environmental health community, or because there are a lot of templates, and I'm wondering.
Yeah, I mean, part of the problem with having a repository for all biomedical ontologies is that it gets overwhelming.
Yes.
And we appreciate that. We have a mechanism within BioPortal to categorize ontologies in terms of various areas of biomedicines. We have some ontologies that are obofoundry ontologies, which is pretty obvious. We have some ontologies that are used in other kinds of projects. And we could certainly create a way of categorizing certain ontologies as environmental ontologies. And I think it'd be better to categorize certain ontologies in BioPortal as environmentally related than to create a whole new repository just for environmental science, because it really becomes really arbitrary as to what's in and what's out of that classification. So yes, we could certainly do that. And that would be a good thing to do. We already have EVO and a number of other environmental ontologies in BioPortal, and I'm sure in the future there are going to be a lot more. Got a question in the chat. Is the CEDAR platform a large relational database or a data warehouse? CEDAR stores its templates in JSON, so you can actually kind of view CEDAR as a big knowledge graph. At least the CEDAR template library is a big knowledge graph. BioPortal stores all of its ontologies and a big triple store. So again, CEDAR BioPortal is also kind of a knowledge graph as well. Some of what's in BioPortal is stored in a relational database. That's mainly the metadata for the ontologies. And so it's really in both CEDAR and in BioPortal, you'll see a mixture of repository types. But that's not something that end users really need to worry about at all. Can you comment on the sustainability outlook for the platform, the level of active feature development maintenance of the core? That's a great question because right now we're in a situation where CEDAR was initially developed as part of the Big Data to Knowledge Initiative, which, as you know, ended a long time ago. All of our work on CEDAR is basically supported by cobbling together funding from a variety of sources, particularly people who are interested in making data fair. We don't have really core funding for the CEDAR infrastructure. That's something we're desperately trying to obtain because right now we're relying on the generosity of projects that know that they need standards adherent metadata to support us, but it's really not the kind of sustained funding that we'd like.
Elaine has a question. Elaine, please go ahead.
Oh, I'm sorry, Elaine.
Oh, no, it's all right. I'm fascinated by the questions that you're getting. And as you answer more questions, it's getting closer to some of the things. So thanks for this presentation. I think it's really, really good. And also the plea for funding. I mean, I think that's one of the things, you know, that actually FAIR principles identified early on is the sustainability of these different efforts, especially through the work through WDS, et cetera. But I did have some questions about functional language. So, you know, especially when we get into the environment, part of it is defining, you know, which environments and so forth. But to get from an effect, an endpoint effect that's well characterized in the medical field, as you describe, or at least increasingly well defined, and an exposure, there's functional and relational language that's needed that is quite specific. I'm wondering, can you talk about that gap in my mind that I see in some of these, you know, current efforts going on?
Yeah, that's a great question. And I think I obviously focused on the idea of standards for metadata in my presentation. And that's really where a lot of our work has been. But there also needs to be standards for data. And I think the kinds of relationships that you're talking about are things that need to be expressed in standardized data sets. And I know NIH has put a lot of emphasis on common data elements or CDEs for various areas of investigation. And I think one of the areas a gap, if you will, so as you say, is CDEs for environmental science. And I think one area where we could partner together for sure is on looking at ways of encoding the individual data as well as the metadata that describe those data.
Oh, I'm so glad you said common data elements because that was one of my other questions. But yeah, that's what we're seeing, especially in the children's health and the epidemiological literature, that we are getting an emphasis on common data elements by kind of focus, you know, whether they're children's cohorts or in utero cohorts and so forth. But there's, and as you know, we've been working on some case studies on this. So anyway, it's exciting. Thank you again so much. You were so organized, and you know, the columns behind you illustrate this sort of organizational approach, even in art. So thank you.
No, that's just Stanford architecture. But I think what you're pointing out is really important. That is, the CDEs and the NIH repository largely were inspired by what were needed for case report forms for interventional studies. And I think a lot more emphasis needs to be placed on what we can do for observational studies. And there's just a lot of opportunity there. Chris asks a question: how critical is CEDAR integration into data repository submission workflows? Are there lessons learned from the collaborations of Dryad and OSF that could be applied across the broader NIH data repository ecosystem? That's a really wonderful question. And I think it's kind of early to answer that question really as well as I would like, because those OSF and Dryad just picked up CEDAR a few months ago. The biggest challenge for both Dryad and CEDAR is that their metadata libraries are fairly impoverished. One of the things that I'm hoping to do in the short term is to prime the pump, get, for example, all of the HubMap reporting guidelines, which deal with a wide set of commonly used biomedical assays in DRIAD and OSF. The biggest challenge, I think, for both of those repositories really is the culture. People have to be able to recognize that we can't just say that putting data in online repositories makes them fair. That's really the misconception that pervades all of science right now. They think if something's online, it's fair. And as I hopefully made clear, it's just showing those ugly metadata records and biosample. That's not the case. We need searchable metadata. We need metadata that allow us to integrate data sets. And I think really the lesson for DRIAD and OSF and all the other generalist repositories is that CEDAR provides a real great opportunity by giving an infrastructure that makes it easy to annotate data sets in a standardized way. And that's the good news. But the bad news is we need the standards. And I think a lot of effort needs to be placed on promoting standards within scientific communities and getting people to use OSF and DRIAD and other generalist repositories that pick up this approach, to recognize that it's really easy to create such standards, to put them into a generalist repository and get people to use them. And I think that's sort of the big win that we get out of DRIAD and OSF right now.
So I don't think we have more questions in the chat. So I have one of my own. And this is something that I don't remember. Is there a feature in CEDAR allowing for to search for an existing template that would be relevant to your data? So instead of coming into the CEDAR and building a template from scratch, when I don't know a name of a template, just to drop several terms like you do in Google and find several existing templates that would be relevant that you can then clone and modify. Is it something that's possible?
Great question. We have a search facility in the CEDAR library, but it is pretty primitive. So you can search for strings and hopefully pick up something. Mm-hmm. What we would really like to do is to basically reformat the whole CEDAR library, because we view that we view the main contribution of CEDAR is that we can take communities of practice and have them store their preferences for metadata in a centralized way. What that means is we need really advanced search capabilities. We need the ability to take primitive templates and expand them to have more options. We basically would love to move to a situation where the CEDAR template library takes on a life of its own as a resource that could be used throughout biomedicine and ultimately all of science as a way of managing metadata standards in hopes of making all of our data sets fair. But we're not there yet. And obviously that would require a lot more work.
Yeah, I guess it comes back to funding, right?
Yeah, so if you know any funders, let me know.
Well, there are some on the call. All right. Let me see. So I don't see any more questions or raise hands. And we're almost at the hour. So I guess this is a good time to thank you very much to our speaker and to everyone who came to listen. And we will meet you again in about a month or so. The next ELK webinar that I have planned so far is about best practices in ontologies. The announcement will come out soon. And thank you and have a good day. Take care, everyone.